As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. Let's zoom down to where this fuel is burned, his cells. Then we'll compare the fuel-burning process in his muscle cells with a very similar process in bacteria. Here, the blood vessel on the left delivers fuel, the black molecules, and oxygen, the red molecules, to a single muscle cell shown on the right. In cellular respiration, energy in fuel is converted to ATP, shown here as yellow starbursts. Most ATP in a eukaryotic cell is made by an enzyme on the inner membrane of the mitochondrion. A bacterium is similar in size to a mitochondrion, and its ATP is formed by an enzyme on the cytoplasmic membrane. This information supports the theory that mitochondria evolved from bacteria. ATP powers the work of the cell, such as muscle contraction or making new cell wall material for bacterial reproduction. Let's take a closer look at how ATP is produced from a molecule of glucose, a common fuel for both humans and bacteria. Only the 6-carbon skeleton of glucose is shown to keep things simple. The first step is called glycolysis, and it takes place in the cytoplasm of both bacterial and eukaryotic cells. To begin the process, some energy, in the form of ATP, has to be invested in order to split the otherwise stable glucose molecule. Now, the molecule NAD+, an electron carrier, shown here in orange, picks up electrons and hydrogen atoms from one of the 3 carbon molecules, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to the electron transport chain, where more ATPs will be made. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced in glycolysis. However, glycolysis has produced 2 3-carbon pyruvic acid molecules, which still have a lot of energy available. Let's follow one of the pyruvic acid molecules into the muscle cell's mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon and 2 oxygens are removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. In bacteria, pyruvic acid is converted into acetyl-CoA in the cytoplasm, since bacteria do not have mitochondria. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that is produced at the end of the last turn of the Krebs cycle. This new 6-carbon molecule serves as the starting point for the next round of the Krebs cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. In humans, this carbon dioxide is exhaled as you breathe. Each turn of the Krebs cycle that begins with 1 acetyl-CoA produces 1 ATP. Since each acetyl-CoA is derived from 1/2 of the original glucose molecule, the Krebs cycle produces an additional 2 ATPs per original glucose. These ATPs are produced by substrate-level phosphorylation. Even more energy is available in the electrons that are being transported by electron carriers such as FADH2, seen here in green, and NADH, shown here in orange. These electron carriers will take their electrons and hydrogen ions to the electron transport chain, where more ATP is made in the process of oxidative phosphorylation. While the Krebs cycle starts another round with the second acetyl-CoA, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion in eukaryotic cells such as this muscle cell. In bacteria, the electron transport chain is located on the cytoplasmic membrane. The chain consists of a series of electron carriers, shown here in purple, most of which are proteins that exist in large complexes. Electrons, shown here in yellow, are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. At the end of the chain, the electrons are transferred to oxygen, shown here in red, and water is formed as a byproduct. The energy released by electrons through repeated oxidation-reduction reactions as they are passed along the chain is used to pump hydrogen ions, the blue balls, across the membrane, creating an area of high hydrogen ion concentration. The hydrogen ions represent a reservoir of potential energy. The hydrogen ions diffuse back across the membrane through a turbine called ATP synthase. Much like water flowing through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in cells produce most of the ATP that is generated from food. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, a bacterium to reproduce rapidly, or it can power your brain cells as you learn challenging microbiology topics. Plants, algae, and some bacteria have another way to produce and use ATP. It's called photosynthesis. Photosynthesis is the process where light energy is converted to chemical energy. Photosynthesis is carried out by plants, algae, and cyanobacteria. These organisms provide us with food to eat and oxygen to breathe. Let's take a closer look at how photosynthesis works in plants, algae, and one group of photosynthetic bacteria called cyanobacteria. In a plant, photosynthesis usually occurs in the leaves. Photosynthesis requires carbon dioxide, shown here as a red and black molecule. Carbon dioxide diffuses into the plant leaf through small pores and then enters the cells. Inside the cells of plants and algae, carbon dioxide diffuses into the chloroplasts, where photosynthesis takes place. Cyanobacteria are prokaryotes that are organized somewhat like the chloroplasts of plants and algae. In fact, they are most likely the ancestors of chloroplasts. Photosynthesis can be divided into two parts, the light-dependent reactions and the light-independent reactions, or Calvin-Benson cycle. Let's first look at the capture of light in the light-dependent reactions. Zooming into a chloroplast, we see these flattened membranous sacs called thylakoids. If this were a cyanobacterium, the thylakoids would be suspended within the cytoplasm. The thylakoids are the location for the light-dependent reactions, where light energy is converted to chemical energy. Let's look more closely at how this occurs on the thylakoid membrane. The two green structures you see here are photosystems, large complexes of proteins and chlorophyll that capture light energy. An electron transport chain connects the two photosystems. Notice the small purple electron carriers that shuttle electrons from one photosystem to another. Now, let's look at the steps of the light-dependent reactions. The photosystem on the left, photosystem 2, absorbs light energy, exciting electrons that enter the electron transport chain. Those electrons are replaced with electrons stripped from water, creating oxygen as a byproduct. The energized electrons flow down the electron transport chain, releasing energy that is used to pump hydrogen ions, the blue balls, across the membrane, forming a reservoir of potential energy. In photosystem 1, on the right, light energy excites electrons, and this time the electrons are captured by an electron carrier molecule, NADPH, shown here in red. As hydrogen ions diffuse back across the membrane, they power ATP synthase, producing ATPs. The light reactions in the thylakoid have produced two energy products, ATP and NADPH, that will now power the production of sugar in the Calvin-Benson cycle. The Calvin-Benson cycle, or light-independent reactions, takes place in the stroma, the thick fluid of the chloroplast in plants and algae. In cyanobacteria, it takes place in the cytoplasm. At the beginning of the cycle, carbon dioxide molecules combine with 5 carbon molecules called RUBP. The resulting molecules go through a series of reactions powered by ATP and NADPH from the light-dependent reactions. 3 carbon molecules known as G3Ps are produced. Most of the G3Ps are rearranged back into RUBPs that will begin the Calvin-Benson cycle again. But the important product of photosynthesis is the remaining G3P sugar. That G3P is combined with other G3Ps to build glucose molecules, which can then combine to form polymers of starch or cellulose. Still other G3Ps form sucrose. And some of the sugar is broken down by cellular respiration in the same cell, using oxygen and generating ATPs that can power other work of the cell. Excess oxygen diffuses out, while more carbon dioxide enters. With 3 simple ingredients, carbon dioxide, water, and light, plants, algae, and cyanobacteria produce sugar and oxygen by photosynthesis. This sugar powers cellular respiration and ultimately provides your fuel as well. Besides cyanobacteria, there are other types of photosynthetic bacteria that carry out photosynthesis in a significantly different manner.